right, well, we'll just jump right into this. I'm having to kind of do a few different things, so sorry if I get distracted because I'm still admitting people through the waiting room. But again, I'm Charlie Doobie. I'm the head of regenerative partnerships here at Agrology. It's wonderful to have everyone here. Really quickly, I'm just going to do some quick housekeeping and that sort of thing. Well, first off, welcome to the Soil Carbon Measurement Strategies webinar. Today, we will be exploring various methodologies for quantifying soil carbon and carbon emissions in agriculture. We're going to be looking at the strengths and the limitations of each method, and also looking at how these various quantification methods tie into and inform carbon farm planning. This is going to be the first of 6 webinars that we plan to host this year, and we'll be exploring a variety of pressing issues in regenerative agriculture, ranging from certifications to measuring and managing soil health, as well as exploring carbon marketplaces, kind of a whole gamut of things. So keep your eyes out for future webinars. Before I introduce the speakers, I just wanted to run through some quick housekeeping. I have everyone set on mute, so please keep yourself muted unless you want us to hear all the fun, background noise of your life. We're going to do question and answer at the end, so you can feel free to unmute yourself in that period. And also feel free to write questions and comments in the chat. I always think it's really fun when the chat becomes a lively discussion, alongside the webinar. So that's there. Feel free to jump in. If you have any questions, we'll try to get to those at the end, and you can feel free to put those in the chat. And then we're also going to follow up with an email to everyone who registered, and that'll include a link to the slides as well as a recording of this webinar. So don't feel any pressure that you need to write everything down, because you will get a copy of all of this. These are the variety of different quantification methods that we're going to go over, soil testing, remote sensing, practice-based models and other models. We're going to be also looking at eddy covariance towers, soil chambers, and the static continuous soil chamber that Agrology has developed. And yeah, without any further ado, I'd like to introduce our speakers. So the first is Miguel Garcia. He holds a PhD in environmental sciences from the University of California at Riverside, and he's the manager of the Sustainable Ag program at the Napa County Resource Conservation District. I asked for a fun fact from each of our speakers, and Miguel shared that when he's not out helping farmers transition towards regenerative practices, you just might find him in your local karaoke bar singing his heart out. He loves karaoke. And our second speaker is Adam Koppel. He's the founder and CEO of Agrology, which is a public benefit corporation dedicated to helping farmers become more resilient to climate change. Before Agrology, Adam has worked as a strategic inventor at various companies, and he holds over 51 patents. His fun fact is that he enjoys both coaching kids soccer as well as getting injured in adult soccer. And with that, I will turn it over to Miguel, who's going to give us an awesome overview of the carbon cycle and kind of take it from there. So Miguel, just let me know when you want me to move slides forward.
Thank you, Charlie. I really appreciate it. And thanks, Agrology, for the invitation. I will be walking you through what the carbon cycle is and why should anybody in agriculture should keep an eye on this, and what the role of carbon is in overall soil health. So here we have a big picture of what the carbon cycle is. Basically, the carbon cycle is the exchange, transport, and transformation of carbon among the Earth's ecosystems. A snapshot of that would be how the carbon dioxide from the atmosphere is taken up by plants, who transform it into carbohydrates that they use as their source of energy. A large portion, sometimes as much as half, of the carbohydrates that the plants produce is actually introduced into the soil. And we call these exudates. These plants' exudates serve as a source of energy for the soil microorganisms. So the plants and the soil microorganisms have a symbiotic relationship where plants provide the microorganisms with a source of energy, and the soil microorganisms assist the plant in accessing resources. So there are microorganisms in the soil, such as the nitrosomonas bacteria, that can fix nitrogen and make it available to the plant. We have other microorganisms that will process organic matter and transform them into forms, transform the nutrients in organic matter into forms that the plant can use. There's other organisms such as the mycorrhiza fungi, which it's another important soil microorganism. It establishes itself within the plant roots and expands through the area. It then searches for nutrients and water, and it will share what it finds with the plant. So this increases the plant's reach. A healthy mycorrhizal community can be extremely helpful when dealing with soils that are depleted of nutrients or when we don't get enough rain. So the plants co-evolved with these soil microorganisms, and they have this symbiotic relationship to keep each other alive. As these microbes process the carbon coming from the plants in the form of these carbohydrates the plant exudates, they incorporate it into their bodies. And when they die, their bodies get incorporated into the soil. These microbes will also digest dead plant material, so that allows to digest whatever plant material is on top of the soil. So as these microorganisms die, their bodies get digested, and then this releases the carbon back into the atmosphere in the form of carbon dioxide. So as the microbes are digesting the organic matter in the soil, plant materials, or the bodies of other dead microorganisms, they're basically doing what humans do, which is we consume food and then we transpire carbon dioxide. So we got carbon dioxide coming into the soil through the plant, and now we have carbon leaving the soil again. So this carbon now could be accessed by the plant again. So hence what we're going against a cycle. So this is what we call part of the carbon cycle. Now, not all of the carbon that is part of the microorganism's body or the plant material or the plant exudates is processed. So some of them gets transformed and it becomes part of a complex, it is processed, and then it becomes part of complex organic structures in the soil. And these structures are very complex. So some of the carbon gets hindered and the microorganisms cannot access it anymore. So these will slow down considerably the decomposition. So by the effect of this, some of the carbon is not accessible. So then it becomes quote unquote stored in the soil. And the longer it stays in the soil, the less it becomes accessible to the microorganism. So we have these two pools of carbon, one that is readily accessible to the microbes, and then one that is not as readily accessible to the microbes. And how quickly or how efficiently this carbon is accessed or absorbed by the microorganisms has to do also with the amount of nitrogen in the soil. So this ratio between carbon and nitrogen will play a role in where having too much nitrogen or having too little nitrogen will impact how the carbon in the soil gets digested. So as the carbon keeps cycling, we see it that it enters the soil, and then some of them will exit through respiration, and then some will get to stay in the soil. And as this cycle continues, we will end up with a buildup of carbon in the soil, because unavoidably, some of it will not be easily accessible to microbes anymore. So this carbon now has been incorporated with other elements in the soil, such as nitrogen and phosphorus, to form stable, complex molecules that we associate with what we call organic matter. So this organic matter is about 50% carbon, but is these organic complex structures that also have nitrogen, phosphorus, and various of other elements. So it is important to note that in this part of the carbon cycle concerning plants and soil, we still see carbon leaving the soil, because the microorganisms are at work, digesting and reproducing. If we would see no carbon leaving the soil at all, that would imply that there's no biology, that the soil is dead. So we want to see this cycling. And in a minute, we're going to talk about the implications when it comes to the greenhouse effect and climate change. So when we are looking at the relation between plants and soils, is about enhancing this carbon cycle. And we want to see the soil generate carbon dioxide, because that tells us that the soil biology is quite active. The more these cycles continue between the plants getting the carbon dioxide from the atmosphere, incorporating into the soil, the microbes digesting it. The more these cycles occur, the more chances for these carbon to become trapped in the soil and be not accessible to the microbes. And we call that carbon sequestration. So when the carbon has gone through these so many cycles that it gets to a point that some of it is just not accessible to the microbes anymore, it just becomes part of the organic matter in the soil. That's what we call carbon sequestration. In agriculture, carbon sequestration is very important because by maintaining adequate amounts of carbon in the soil in the form of organic matter, we ensure a healthy biological environment where plants can thrive. The carbon in the soil will also influence the chemical and physical properties of the soil. For one, the organic matter stores nutrients that become slowly available as microbes digest it. So it is a continuous slow release of nutrients for the plants. And organic matter also serves as a glue that keeps soil particles together. And this contributes to a stable soil structure that promotes good soil porosity. This helps enhance the ability of the soil to retain water efficiently. The carbon cycle obviously goes far beyond agriculture. Humans, of course, are a great source of carbon in the form of carbon dioxide through the burning of fossil fuels. Forest fires are also a great source of carbon. And this carbon dioxide builds up in the atmosphere. And carbon dioxide is a greenhouse gas. So having excess carbon dioxide in the atmosphere enhances the greenhouse effect. This contributes to global warming and eventually manifests in the form of global climate change. Carbon dioxide can be dissolved in the oceans and either stay dissolved in the oceans or be incorporated into coral in the form of calcium carbonate. So this is a form of inorganic carbon compared to the organic carbon that we were talking as part of organic matter. Calcium carbonate can also be found in soil. So we have forms of organic and inorganic carbon being present in the soil. So in the oceans, it's helping build coral. However, even though the oceans have a great capacity to store carbon, we're getting close to the limit where now the additional carbon being added is actually acidifying the oceans, and instead of helping build more coral is preventing. So it's slowing down the buildup of coral. So coral that gets damaged doesn't get to regenerate as easily. So between global climate change and ocean acidification, among other global concerns, we seriously have to think about ways of reducing the amount of carbon that is entering the atmosphere. And soil happens to have one of the largest capacities to store carbon. According to scientific estimates, soil can store as much as twice what the atmosphere could if it is managed properly. Agricultural soils in particular can be managed in a way that enhances carbon sequestration, which can help reduce the amount of carbon in the atmosphere while enhancing soil health. So it's a no-brainer. The more carbon we can put and keep in the ground, the more carbon can be removed from the atmosphere, and the better our soil health will be. So when farming is done in a way that optimizes carbon capture, we call that carbon farming. It is a strategy for achieving carbon storage and carbon sequestration by implementing specific conservation farming practices. And a strategy to achieve that is called a carbon farm plan. So Charlie, if you could please go to the next slide. Thank you. So developing a strategy in which a farm will be enhancing the amount of carbon that gets into the soil and that gets stored into the soil is called a carbon farm plan. So this is a carbon farm plan incorporates a whole farm approach to enhancing the rate of carbon sequestration through the implementation of conservation practices that enhance this carbon cycle. So we want this carbon cycle to continue, and we want the biology in the soil to be active, but we want to make sure that as much of that carbon stays in the soil to enhance soil health. The Napa RCD has been developing carbon farm plans for vineyards for several years now. So please contact me if you're interested in learning more about the process, if you're interested in having a carbon farm plan developed for your vineyard. Our neighboring RCDs in Sonoma and Mendocino also develop carbon farm plans. So if you're farming in these areas, feel free to reach out to them. There's a lot of activity related to carbon farming around California and in other parts of the country and other parts of the world. So anywhere you are, reach out to your resource conservation districts, reach out to the USDA to see if they can assist you with developing these carbon farm plans. In our website, naparcd.org, again, naparcd.org, we have a sample of a carbon farm plan that we have developed recently, in case you're curious on how the plans look once they're completing. Next slide, please. In the carbon farm plan, we describe the carbon sequestration potential of various conservation practices. So we talk about, I just have a couple examples here: cover crops, mulching, and reduction in tillage. Cover crop will enhance carbon sequestration because the living roots are themselves exuding carbon, and that is contributing to the biology. Mulching conserves moisture and also helps enhance the biology. I'm going through this in a very generic way, but if anybody's interested to learn more about it, you can always reach out to me. In the case of no-till, the reason we promote no-till or reduction in till as a carbon sequestration strategy is because when the soil is left alone, everything is in equilibrium. So within the soil, you have areas with oxygen, areas with no oxygen, but everything is in equilibrium. The moment we disturb the soil, we expose the more surface area of the soil, and by default, we're exposing more of that organic matter to the oxygen. So some of it gets oxidized, so you can lose organic matter just by oxidation through exposure to the air, but also you're going to have a disturbance in the balance of the amount of oxygen that is entering the soil, and that is going to alter the biology in the soil, since we have microbes that like oxygen and microbes that don't like oxygen. So by leaving the soil alone, you allow the soil to reach its natural equilibrium, and by not exposing a large surface area of the soil to the air, you're preventing some of the oxidation of the organic matter. Again, I'm going through this in a very superficial way. In the current farm plan, we need to quantify in order to, we need to quantify the amount of carbon that these practices could sequester in order to develop a strategy and to be able to prioritize, but also programs, state programs such as the Healthy Soil Program will pay you based on estimation of how much carbon you can sequester. So we use in our current farm plans the Comet Farm and the Comet Planner tools, which is a model that was developed by the USDA in collaboration with Colorado State University. And this gives us metric tons of carbon dioxide that would be sequestered if the conservation practice in question were to be adopted. So if you tell me I have 100 acres, I've been tilling regularly, what would happen if I stop tilling? So this tool will give you an estimate of how much carbon you would be preventing from going to the atmosphere and how much carbon would stay in the ground if you were to reduce your tillage by acreage. Now, this is a model, so it is based on several peer-reviewed research papers, but it is a model. So it is not perfect. It is one of the best tools that we have available to make a prediction, but it's not perfect by no means. So the best way to really know how these practices are sequestering carbon, how much carbon is sequestering, would be by actually measuring the amount of carbon in the soil. So if you were to actually measure the amount of carbon that is in the soil before and after you implement any practices and throughout the years as you're implementing practices, that would be the best way to really know what's really happening. But when we're trying to prioritize what could have the greatest impact, this tool is very useful just as a mental exercise so that you can see, okay, if I have limited resources or limited time, what can I do that would have the greatest impact. And again, it's not perfect, but it is a very useful tool. It is very powerful, and it is scientifically based. It's based on scientific peer review research. So, and it is constantly being updated. It is constantly being refined. So it is constantly becoming better and better. So we use these tools to develop a strategy when we're developing compound plans. Next slide, please. So, like I said, the best way to know how much carbon you are actually sequestering or how much carbon you have to begin with would be by sending soil samples to a lab. So this involves sending somebody out in the field and collecting several soil samples. The method that we use, or the method that we suggest people use, is what the USDA refers as the aggregate sampling, meaning you pick an area. Let's say you have a 20-acre plot. You would go and collect about 10 samples throughout those 20 acres. All of those samples get put in a bucket that is mixed thoroughly, that is put in a bag like the one shown in the picture here, and then that gets sent to a lab. The key is that you need to make sure that you have a very thorough representation of your field, in that every sample is collected at the same depth and that you're collecting the same amount of soil. Because if you go and collect more from one region than the other within your sampling area, that could also keep discrepancies. So the benefits of this method is that you develop an average of what that field looks like. And ideally, the entire field should have the same soil texture, same slope, all of that. But if it doesn't, then those become different subsections that you need to sample. If there's significant differences in the soil type, you need to make sure that you sample those separately to make sure that you get good representation. And then make sure that the same person is doing the sampling, or that everybody in your team knows the protocol and that everybody's following properly. So again, make sure that you have a good representation of the entire field. Make sure that the soil is collected at the same depth, and make sure that the same amount of soil is collected in each of those sampling sections, and make sure that everything is mixed very thoroughly before sending the samples to the lab. So when it comes to carbon and what the labs can tell us, there are different methods that will give you different pieces of information. So I'm going to walk you through some of the most widely used methods for measuring carbon. So the first one would be soil respiration. So you will send a sample to a lab. What they will do is they will dry the sample and then re-wet it. And then they will put it in a chamber where they're measuring the amount of carbon that is coming out of the ground for the next 24 hours. So this is a sample that came from your property, was dried, air dried, and then re-wetted. And that re-wetting, essentially what it's doing is restarting the biology in the soil. So all the microbes that went dormant because there was not enough moisture for them to become active, all of a sudden become very active, and they measure the amount of carbon dioxide for the next 24 hours. So what that is telling you is basically how biologically active your soil is. The drawback of this is that it's done in a laboratory setting. So you're taking the soil that was disturbed, you're taking the soil that was sent out, and then you're re-wetting it. So you're getting a pretty good idea of what the biology of your soil is. And as long as you're repeating the same method, you can compare between seasons, or you can compare between sections of your field. But it is based on artificial conditions, quote unquote. Still very valuable. It still gives you a lot of information. Another method to measure carbon in the soil would be, let me just take a step back. So for the soil respiration, it is essentially measuring the biology in the soil and how much organic matter is being digested. So those are the microbes taking the organic matter that is already existing in the soil and then digesting it. So that's telling us also the ability of the microbes to digest what is already there. So it is sort of like an indirect measure of how much carbon you have in the soil. If there's no respiration, that means that either you don't have enough biology because there's not enough energy source for them. So that can tell you that you're depleted of organic matter. The next method would be total carbon by loss of ignition. So this method is trying to measure all of the carbon that is in the soil. And what they do is basically they introduce an amount of the soil into a furnace. They keep it at 360 degrees Celsius for about 2 hours. They weight the sample before and after. So this has to be dry, of course. So you have to remove the moisture first. Then once the soil is dry, they put it into the furnace and they burn it at this temperature, trying to basically release some of that carbon that is incorporated into the soil. So then they measure before and after. This would be probably one of the cheapest methods out there because they basically just put it into an oven, and then you can do these probably with your, if you buy a furnace yourself. They're not, it is not a fancy lab equipment. So if you send a sample to a lab to do this, should be relatively inexpensive depending on what is around your area. However, at that temperature, depending on the amount of clay that you have there, depending on the accuracy of the instrument, how long you keep it in the furnace, you may or may not get a true representation of how much carbon really is in the soil. So the solution to that would be to actually combust the entire sample. So that's the next method, which would be total carbon by combustion. So in this case, the carbon, the whole sample is burned to the ground. So it's burned entirely. And this is attached to an instrument that would measure the amount of carbon dioxide coming off the sample. So everything in the sample is burned to the ground, and the instrument is directly measuring the amount of carbon dioxide. It can also measure the amount of nitrogen in the same way, because you can measure the amount of nitrogen gases that are released. This is a more expensive lab instrument. So if you use this method, it would be a little more expensive, but it's more accurate than the loss of ignition, because you're actually burning everything down in measuring the amount of carbon dioxide that is coming out of it. One thing to consider here is that the carbon dioxide that is being measured is everything, all the carbon in the soil. But this also can incorporate the inorganic carbon that does not form part of the organic matter in the soil. So if you want to know just the organic matter, the samples need to be, they acidify them, so they add acid to remove the carbon that is inorganic. So any calcium carbonate or any other carbonates in the soil can be removed by acidifying. And so this is something that the lab should know. Just know that if you just ask for total carbon combustion, it would be all the carbon, organic and inorganic carbon in that sample. Then finally, the last method to look at carbon in the soil would be by the permanganate oxidation method. So this called, when we measure these, we're measuring the potential carbon in the soil that can be oxidized by potassium permanganate. We call these POXY for short, potassium permanganate oxidizable carbon. So this is carbon that gets exposed to a very powerful oxidant, which is potassium permanganate. And it's essentially measuring how much of this carbon can be oxidized by this high strong oxidant. And what this tells you is the portion of the organic matter that is active for soil microbe activity. So meaning the portion of the organic matter that is easily accessible to the soil microbes. So we talked about earlier how there are portions of organic matter in the soil that are just hindered in complex structures that the microbes cannot access anymore. So that's the carbon that becomes, that stays in the soil for a longer period of time. And then we're going to have the portion of that the microbes can actually access. So this method would only tell you how much of that carbon is actually available to the microbes. And this is important to know because this will tell us the amount of energy available for microbes to access and for the microbes to be able to do their job. So all of these are ways of measuring carbon in the soil. They're measuring different things. They're telling us different things. One of the drawbacks to these methods is that you get a snapshot in time of when you collect the samples. The carbon cycle is always active, but the rate of carbon sequestration will be different throughout the season due to weather variations, soil moisture, and soil management. It can also be different from season to season due to these factors. So when you're collecting these samples, you're just getting a snapshot in time. So you have to be very careful on when you're collecting the samples. So if you want to know what's really happening throughout the year, you would have to collect samples at different points in time during the season. That is time consuming and it can get expensive. So we would have to decide, when is the best time to collect the samples. And then if you want comparisons from year to year, you would have to make sure that the samples are collected around the same time, samples are collected about the same way, and that they're sent to the same lab. Because unfortunately, these type of analyses are not regulated. So if you send your sample to 10 different labs, you might get 10 different results. Not to take away any credit from how valuable these methods are, but the reality is that it can vary from lab to lab. So it's important to stick around with the same lab and make sure that everything is pretty much the same. But the reality is that you're just getting a snapshot in time. So you're not going to be able to see what's the subtle differences that are happening from season to season. Forget about seeing what's happening throughout the day, because then you would have to collect samples, at different times during the day. It's not practical. So with the lab analysis, we can get a lot of information. Just keep in mind that these are limitations on the kind of information that you can get. Next slide, please. So just to circle back, not pun intended, into the carbon cycle and the importance of it in agriculture. So it is important to know that carbon sequestration is an effect of the carbon cycle. The carbon cycle is just constantly moving. It's constantly changing carbon from the atmosphere into the soil, back into the atmosphere, and it's going through these cycles. Now, the more of these cycles and the healthier these cycles are, the more the chances that some of that carbon will become unavailable to the microbes or will become less easily accessible by the microbes. So we want this cycling of carbon to be happening in our soils, in agriculture and forests and anywhere else. And the soils that are the healthiest are the ones that are going to be the most active. So you're going to see more carbon coming out of the ground in healthy soils. Now, this sounds counterintuitive because we're talking about we want to keep that carbon in the ground. But it's important to know that the more these cycles are happening, the more this carbon is becoming sequestered and the less that is accessible. So the more carbon dioxide is going in out of the soil through this respiration and how the microbes are just doing their work, digesting and reproducing, the more carbon that would be entering the system. So we want to see these carbon fluxes occurring in healthy soils. And the more they happen, the more carbon will eventually become stable in the ground. And that will contribute to removing carbon from the atmosphere. And that has a huge potential to mitigate the impacts of climate change. So when you walk in your farms, I encourage you to look down because you are walking in an immense universe of life. In one single tablespoon of soil, healthy soil, you can find as many organisms as humans have ever lived on Earth. So there's a whole universe below ground, and the more we take care of it, the better it will take care of us. That's all I have. Thank you.
Thank you very much, Miguel. And now we'll move to Adam, and he will look at, we'll jump into some of the other methods of soil quantification.
Yeah, and so I don't want to be too repetitive here, since Miguel just did a great job of explaining the pros and cons of soil testing. And so we'll go real quick on this slide before I move on to the others. So to emphasize again, if you really want to understand soil carbon, the absolute ground truth is measuring the soil. But of course, it's really hard to do that at scale because you need to physically sample each place, and you need to blend samples across depth. And then you need to make sure the lab is doing a consistent procedure on a properly calibrated machine. But this is a very widely accessible and extensively supported approach. Without a ton of upfront cost, you can get started with it very cheaply. And the challenges with it really come down to the fact that you have snapshots in time. There's variation between labs or probably even machines doing the analysis within a lab. Not everyone's out there sampling their soil every day. And another thing I'll highlight, of course, is that this does miss other greenhouse gases like nitrous oxide, which are particularly potent and cannot be captured in a soil test analysis. So without spending too much more time on this, let's move on to the next slide. So another approach to assessing soil carbon is remote sensing. And remote sensing means measurement from afar. So as opposed to getting down in the dirt and actually measuring, you're using sensing equipment typically mounted on an aircraft or more frequently a satellite to gather data to understand what's happening. And there's some big advantages to this approach, specifically the fact that it can cover a wide area and it can do it very quickly. So, there are satellites that essentially scan the surface of the earth almost every day now. So you have an enormous data set getting built up, and that happens autonomously. There's minimal labor involved. For aircraft, there can be, or for drones, there can be, but these satellites are flying all the time up in orbit, gathering that data. And so it can be a really powerful way to understand what's happening. There's some significant cons too. First off, there is low resolution. So especially as we look at specialty crops, where plots and blocks are small, it can be hard to say, this area here, this 5 acre plot that's cultivated differently than neighboring 5 acre plot, what's happening there. It's just inherently a low resolution technology. The second challenge here is it's low accuracy. So remote sensing tends to be really good at relative comparisons. So you might be able to say block B has higher soil carbon levels than block A, but you can't say what either of those soil carbon levels are. And that's challenging. Additionally, it typically excludes non-carbon greenhouse gases without making some guesswork involved. So this is not a way to approach nitrous oxide emissions. There are some aircraft and drones that will fly over with gas sensors to measure gas flux in a wide area. And that's an effective way maybe to look at some of this, but typically that's not considered remote sensing. So I want to call that out. And then finally, there's minimal scientific track record here. So there have been a few papers published that cover remote sensing for carbon, soil carbon analysis, but not to the degree where it's a widely accepted methodology in the scientific community. And that can be challenging when you're looking at a measurement approach and you really want to build the credibility of the performance and outcomes of your carbon farming plan. Next slide, please. So the next approach is carbon models. And this is a widely used approach because it was one of the state-of-the-art things for a very long time. So we've already mentioned COMET. There's also the DNDC model. And finally, there's the Cool Farm tool. And these approaches have a number of great advantages. First of all, they can be very low cost or free. So they're a great way to start experimenting with carbon monitoring on farms. They're very accessible. Some of these tools have web-based interfaces or app-based interfaces that make it very easy for farmers to go in and start understanding this. They are generally accepted as a way to quantify the benefits of various carbon farming practices, and that's great. And they, fundamentally, they're a lot better than nothing. And that's a really important thing to take away as well. That if this is the only approach, and it has been for a long time, it's great. And so especially if you do modeling, maybe combined with some soil testing, that's an excellent way to get an understanding of what's happening on your farm and to show that your carbon farming plans are having outcomes. There's one major con here, and that is that models are not measurements, and they never can be. So this is all hypothetical, saying if you hypothetically make these changes to a hypothetical farm in this hypothetical area, here are the hypothetical outcomes, and they cannot be guaranteed to apply to your farm. These models are typically built for academic purposes to understand, broad ecosystems, but what happens in a model doesn't necessarily happen exactly on your farm. And so that's these measurements and monitoring and suggestions. The models need to be taken with a grain of salt. Next slide, please. And now we start moving up the difficulty and cost structure. Eddy covariance towers are a greenhouse gas flux analysis approach. And so what an eddy covariance tower is basically a 3-dimensional sonic anemometer that measures wind speed, and then a bunch of gas concentration analysis equipment, and then a lot of computation to basically measure over time what the net flow up or down out of the soil and the plants are of greenhouse gases. And so you can probably already tell from my description that this is not a cheap approach. And before I get into the cons, we'll stick to the pros here. So one great thing about this approach is it's real time. the data streams out of these towers in real time, so you can see how cultivation practices, weather events, seasonality, all of these things are affecting the sequestration or emissions of carbon out of the soil. They also have pretty good sensitivity, so you're sensitive to a lot of different kinds of events. They are pretty accurate, so you get a really good real-time register. These devices or these towers can typically look at more than just carbon. So they can look at carbon and methane and even energy exchange through temperature and H2O to understand energy exchange in the ecosystem. They're widely regarded as the scientific gold standard for understanding the flux or the flow of greenhouse gases in and out of a natural environment and agricultural environments. They also cover a moderate measurement area, which is nice. So it's not just the footprint of the tower can be a few 100 meters radius or even larger depending on the height. So there's a lot of really excellent advantages to this, which is why they are used extensively by researchers in scientific environments. But then there's some cons that make them difficult for applications in practicing agriculture. The biggest con is the extremely high cost. So these devices will cost on the low end tens of thousands of dollars, on the high end hundreds of thousands of dollars. And it's unfortunate because they're great devices, but it's just going to limit their applicability. Additionally, there's practice compatibility issues. So if you have, let's say sprayers or trimmers or any equipment like that running through fields, and you have an eddy covariance tower in that field, and that eddy covariance tower gets hit by the sprayer or trimmer, that's a pretty big problem. They also can be vulnerable to weather potentially, and just a lot of challenges like that because they're inherently expensive. They'll require some maintenance, which is challenging because that maintenance typically requires a pretty qualified technician. They are also not very mobile, so the footprint that they measure is pretty fixed. You can't pick these things up and move them. And they have the potential to miss some flux during still air periods because fundamentally this technology requires movement of air to measure the flux. However, these cons don't outweigh the pros. The real challenge here, we'd be using eddy covariance towers everywhere, is that cost barrier. And also the aligned practice compatibility area. If these things were cheap and durable, I think we'd see a lot of application, which would be awesome. Next slide, please. So an additional greenhouse gas flux approach is the soil chamber. And so the soil chamber is essentially an enclosed volume where concentrations of gases are measured over time to see if the soil is emitting them or absorbing them and in what rates. And there's a number of pros to this approach. It is real time if it's a digital analyzer connected or inside. It's highly sensitive, so you can pick up minute fluxes of gas in and out of the soil. It's highly accurate because the sensing technologies used to measure this have been refined over decades. And you can pick up carbon and other greenhouse gases, including methane and nitrous. And then finally, there's an extensive scientific track record. There's almost 10 decades of lineage of soil chamber research, starting with physical manual sampling, and then extending all the way through the automated systems we say today. And actually another pro I forgot to mention here is that when the sampling is done manually, a soil chamber can be practically free. You can build one out of components you'll find around a junkyard, and you can measure gas files and send them to a lab for analysis via the manual process. But the challenge, of course, is always that's manual. On the con side, these systems that are automated are extremely expensive, similar to eddy covariance towers. There's also the labor of those manual systems if you want to get a lot of data over time. The automated systems can produce continuous data, which is great, but those manual systems, unless you have a huge capacity of affordable labor, you're not going to get that much data out of them. We're going to run into practice compatibility issues again on actively cultivated farms. These devices, especially the automated ones, can be very expensive, and you don't want to be hitting them with tractors or mowers or sprayers. They also measure fundamentally a small area, which is the area directly on the chamber, which is challenging because if you haven't selected the right area of soil to measure, you're not sure that's representative necessarily of the field. And then finally, a lot of these devices open and close as part of their operation to flush the gas. And while that's advantageous for ensuring that it's measuring well, it can be a problem because there's actually transient signals that you can measure from the soil. And depending on the open and closing cycle, those transient signals may be missed. And so now we'll go on to the next slide. to talk about Agrology's approach for greenhouse gas flux analysis. And so we looked at all of these measurement approaches. We tried to find a way to get the best of all of them without a minimum amount of downsides and provide growers with the continuous feedback in rich detail that will really help them both with managing soil health, measuring soil carbon sequestration, but also just agriculture in general. And so we saw that the soil chamber was a nice way to be highly accurate on carbon leaving the soil as well as nitrous. And that it'd be great if we could get something like an eddy covariance tower without the cost. And so we combine a soil chamber with a concentration gradient mask that measures CO2 concentration at different heights around the canopy. And this allows this approach to continuously monitor and quantify soil carbon flux. And eventually, we believe, net ecosystem exchange. It's also a great approach for gathering real-time insights on soil health, since we see soil respiration happening continuously. It lets growers see the effectiveness of the regenerative practices very, very quickly. The device itself is small and easily mobile and can be installed or uninstalled within a minute, which is really important for two things. One, it allows the device to be moved around an agricultural operation between different blocks or different fields or even different rows, just to capture more data so you can be more confident you're getting represented data. And additionally, it lets you remove a device if agricultural practices and cultivation practices could interfere. So for example, if you are a tractor operator tilling a field between vegetable plantings, you can pull this device out as you approach it and reinstall it behind the tractor's implement in just a couple of minutes with minimal disruption. There's no labor beyond the installation and movement of the device. And then finally, it is a multi-greenhouse gas capable device. Next slide, please. So one of the great things about this soil chain approach is the rich real-time carbon data. And so we see here some data from an agrology test site, where first we see the effects of rainfall on the far left. So precipitation actually fills the soil pores, flushes out the CO2 in the soil pore, and we're capturing that in the arbiter device's chamber. Over time, we also see growing significance of the root exudate triggered microbiome respiration spikes. So what's happening here is as photosynthetically active radiation arrives on the plants, both the cover crop and the vines, that triggers a release of root exudate in the morning to stimulate energy exchange between the soil microbiome and the plants. And we see that energy exchange happening with a burst in activity in the soil. And so that's a fun thing we've realized is actually a really great way to track soil health in real time, similar to a vital sign of the soil that you can measure electronically. And then additionally, we'll see the diurnal soil carbon respiration cycle with differing levels of activity between night and day. Next slide, please. Additionally to seeing these things, it's applicable to understanding carbon farming and regenerative agriculture. So we have here a data set showing on the same area, about 30 rows apart in a vineyard, a conventional block and a regenerative block. And the thing that's so awesome is that the data is visibly different. The conventional block has minimal root exudate bursts in the morning, and the conventional block is minimal root exudate bursts in the morning, and the regenerative block tends to have massive ones. And so just as we heard from Miguel earlier, you can really see that this regenerative healthy soil is breathing and cycling carbon exactly how we would expect and hope to see. And this is real data. This isn't anything that's been faked or mocked up. This is the raw CO2 concentration stream from that agrology arbiter chamber. And it's just instantly apparent how a healthy soil respires so much more carbon than a conventional. Next slide, please. We can even see this in the flux analysis. So here we're now moving for the same regenerative and conventional block. We're looking at the carbon flux. And the fascinating thing here is we see for just the chamber aspect of this component that the regenerative block is respiring very vigorously, cycling CO2 back to the canopy. And so we see a lot of carbon dioxide coming out of the soil. And that is counterintuitive, of course, because we want to see sequestration in the soil, but healthy soil with sequestering carbon is going to respire a lot more CO2. That conventional block, fascinatingly, actually absorbs CO2 occasionally. And we believe that's due to a lack of in-pore availability of carbon dioxide and the soil needing to absorb it to support any level of biological activity. So again, sometimes when you measure this data, you'll see things that are counterintuitive, but are explained once you understand the role of healthy soil and how carbon cycles. Next slide, please. And the final slide we have here is arbiter data, again, from the conventional and regenerative difference, now looking at canopy analysis of CO2 concentration in canopy. And again, we see the conventional blocks tend to absorb less CO2 than the regenerative blocks, which maintain a lower level of carbon dioxide, a lower concentration. And that's exactly what we'd expect to see, where the healthier soil is growing healthier vines that are absorbing more CO2, releasing that as exudate into the soil, and fostering a healthier soil that is then respiring more carbon back up. And so, all of this data can confirm hypotheses or studies that you see in scientific journals, but now you can do it actually on almost any farm by deploying this equipment. And the great thing here is this is all real-time data, which lets growers connect their practices to outcomes very quickly, typically within just a few weeks. So next slide, please. That's what I figured. I think we're now at the question and answer portion. I've seen questions coming in. And Charlie, I'd ask if you don't mind, please, to start feeding those to us so that we can answer them.
Yeah, sounds good. So we had the first question about remote sensing and can it be accomplished through low-flying drones? And then Michael had a great answer there saying, yes, they can do it, but like satellites, they will not measure the fluxes. And they will give a summation of the industrial, field, and livestock emissions, but there's no way to separate out those different, methane sources. And then he also said, highlighted that again, that would be a temporary snapshot. So it's not going to be ongoing flux. I think that's a great answer. So we'll probably just leave it at that. And then Lee asks the question, can you review the pros and cons of sensors like Teralytic that measures oxygenation and CO2 respiration rates at 6 and 12-inch depths? Are you familiar with that, Miguel? Or
yes, so I'm familiar with Teralytic, and apologies. The question was, what is the pros and cons of that approach to measuring oxygen in the soil?
Yep, oxygenation and CO2 respiration. And also, Lee, if you want to take yourself off of mute and you could, kind of jump in a little bit deeper in that question.
Yeah, you have done such a great job kind of going over the other aspects that, I was kind of curious just in terms of, maybe some of the below ground soil sensors, just kind of what your experience has been with them in terms of accuracy of the data, because I've kind of seen some anomalies with some of the no-till and low-till stuff where kind of some of that CO2 respiration rates actually decline at 12 inches below the surface where things are getting very healthy and are kind of becoming more active in the shallow soil, but I don't know, I imagine kind of nutrients are kind of being pulled up, but I was just kind of curious, of, what's the value of measuring at the surface versus measuring below, and have you kind of come across sensors that you feel are generating accurate data?
Sure. Okay. Thank you for that expansion, Lee. So the first thing I'd call out here is, I haven't physically analyzed Teralytic devices. I'm not aware that they're in market yet, and that's always a challenge. So if they are, and anybody has experience there, please participate. I've only heard that they are in a kind of prototyping phase and haven't been widely released. So that always makes it hard if the product isn't in market to speculate on the pros and cons. So the challenge to measuring oxygenation and CO2 respiration at depth is that that's a snapshot in depth. And I think for oxygenation, it's a great way to understand if the roots of cover crops and primary crops have access to oxygen in the soil, which can definitely promote healthy soil. But those approaches will not get you to understanding the flux of gas in and out of the soil, which is really the ultimate goal here for carbon farming, is to understand, is the soil healthy? Is it emitting CO2 and sequestering CO2 as expected? Are we getting towards a net ecosystem exchange understanding of the carbon in versus carbon out of the soil? Just measuring in the soil isn't going to be enough to do that. And that's just essentially physics, because you're not looking in the canopy and you're not looking at emissions. So, what I'd say is at Agrology, you're always excited to put out new sensors with new applications. And This is certainly something we'll look at to see if we can get data on CO2 emissions in the soil, but ultimately by measuring at the surface, we're capturing the entire gas profile of the soil underneath the chamber, which is advantageous as well. Does that cover your question, Lee?
We'll assume that it did.
All right, great.
Yeah, thanks for that, Adam. Okay, so the next question is, how much carbon is sequestered per acre per year from cover cropping, mulching, no-till, and reduced till? And I'll turn this to Adam or Miguel, but I'd say first and foremost, it's just important to highlight, it really depends on soil characteristics, on location, on the management history of that, so there's no easy answer to that. Miguel, I don't know if you want to jump in. I have some numbers that I've seen in terms of averages there, but yeah, first and foremost is it varies, is I think the easiest.
Yeah, I think you got it right. But if you can show some numbers, just so that people can get a reference point, I think it would be good. But when you go into the Comet Planner model, you are asked to put in your county. So the model is tuned in for different areas of the country. And California has their own specific version of it as well. So when you put in your county in California, it will give you an estimate of what it would be expected for where you are based on your soil characteristics, based on climate. But the reality is that it would depend really on where you're farming. Again, it's a model, but the model does incorporate weather and region into it. Would you mind, Charlie, sharing some of the numbers that you have seen, just so somebody can get a reference point? And then just keep in mind, whatever you hear from Charlie, it can be plus or minus a specific amount.
Absolutely. So there's this really interesting study that I saw that it was a meta study. So it looked at, I think, around 80 or 90 different peer-reviewed studies that looked at it specifically no-till and cover cropping. The average, and don't quote me on this, and I'd be happy, I can send this this study out because it is an interesting one. So in the follow-up email, look for that. But for cover cropping, I believe it was around 3/4 of a ton per acre per year. But what's really interesting there is that around 10% of the fields that were studied actually had less carbon sequestration after they moved to that new practice. And then around 10% also had over 2 tons per acre per year. So that just shows there's a wide variability, all the way from actually a negative impact through the average of around 3/4, all the way through, 2 tons. And then it was similar numbers for, so that was no-till, similar numbers for transitioning to cover crop. And I believe that was a little bit lower than no-till, but it was around like 0.67 per tons of carbon dioxide per acre per year. So I'll follow up with that study and you can look into it. And again, I think what's really important to highlight there is that if you're using these practice-based models, it's not going to look at that wide range. That's where, actually measuring what's going on in the field is really important, because depending on a variety of factors, you could have a wide variety of different outcomes. And so it's important to measure the actual outcome of your practices.
Another thing to consider is that the amount of carbon sequestration would change over time. So if you're beginning with a very healthy soil that already contains a good amount of organic matter, the amount, at that point, you're not sequestering anymore, you're maintaining what you already have there. So the initial conditions, how much organic matter you have to begin with, will also play a role over time, and how long you implement the practices. If you're no-till for 10 years versus 5 versus 2, that's a difference. Something that is so up in the air is, for those folks that like to do alternate tilling, meaning they till some sections some years and then they rotate them with other sections of the years. Is that enough time to sequester carbon and keep it in the ground, or are you removing all of the carbon that you sequester? It really depends on the site. I've seen research papers that talk about losing as much as half of the carbon that you introduce, and other research papers showing that not as much carbon is lost, because it has to do with the intensity of the tilling, the moisture of the soil. So there's a lot of factors that will play a role into the rate of carbon sequestration that these models just can't capture, because there's just so many moving parts. But ultimately, what we have seen is that the longer you implement these practices, the better carbon sequestration you would see over time, and then your soils would reach a capacity. So all soils would have a capacity. Clay soils tend to retain more organic matter than sandy soils, so you can't expect a sandy soil to perform the same way that a clay soil does, and there's variations in between. So getting to know your soil is very important for you to determine what the limitations are as far as carbon sequestration goes.
So it is 11:03. So I just wanted to highlight that. If people have to start jumping, we appreciate you coming. Really appreciate all the questions. We'll stick around for a bit, and we'll continue to ask and answer some of these questions. We will follow up with an email with the recording, with the slides, with that study that I mentioned, as well as everyone's contact information that was asked for. So I'm just going to jump to the next slide here, which has some contact information for both Miguel and Adam. Again, I will follow up with an email, so you'll have my email, but just wanted to highlight that we're at the top of the hour here and really appreciate everyone coming. Again, we're going to be having more of these webinars, but I think that fundamentally coming together as a community and having these conversations is super important. So we're excited to play our part and would be excited to talk to anyone who's interested in exploring these topics further. So yeah, to jump back to the questions, our next question was around the costs of the agrology devices. And then it says, what are the O&M costs? I believe you're probably saying ongoing maintenance costs. Maybe you can expand on that if that's not the correct interpretation. But Adam, do you want to talk about cost structure?
Yeah, sure. And so the challenge here, of course, with any cost thing, it depends on the scale. So for the smallest range of work that agrology will typically engage in, it's going to be about $7,000 a year for a few devices that can cover about 60 to 80 acres. That includes the devices, all the analysis, the maintenance, the warranty, and typically installation and movement is necessary. And so that's kind of the starting point. Comes down quite a bit from there as costs improve. And ultimately, what we hope to do down the road is connect with carbon markets to help offset some of that cost through the creation of high quality, continuously monitored carbon credits. What I'd say is if you're interested in the costs and understanding how they can be applied to your project, to please reach out to us and we'll be happy to discuss it and figure out how we can come to an arrangement that works. We've collaborated with a lot of academics to help validate the product. If you're coming from an academic environment and can help us with that, we're always eager to do it. And then we're open to exploring how to make this work for other agricultural systems and other types of companies. So please don't let that initial cost discourage you. Let's get on the phone and talk.
Awesome. So the next question, well, I think it's important to highlight just looks like Damon. I'm not sure if I'm pronouncing your name correctly, so apologies on that, but was highlighting that they were able to sequester around 10 tons of CO2 per acre per year in a Virginia farm. I would highlight that the numbers that I was throwing out there, that is a change in sequestration. So that's not total sequestration, but that's a change based on a practice change. Additionality is, a big thing in the carbon space in terms of looking at if there's a change in practice or a change in the structure, what is the additional carbon that is sequestered thanks to that? What is the additional impact? So yeah, I just wanted to highlight that. And then we have a question from Michael around the spike pattern that we associate with the root exudates. He says the spike pattern you associated with root exudates are highly unusual in biology. Can you elaborate more on the mechanism of action and how you validated this pattern with an independent technology, and also at what resolution this happens?
And that's a great question. I want to highlight to everyone that Dr. Michael here is a foremost expert on gas flux and soil chemistry and a range of other subjects. And it's great to have you on the call and to talk with you again. And so if we could actually go back a few slides, please. If just so we can have some visibility to come into this.
This one?
When this first occurred, yeah, that's perfect. When we first saw this pattern, we thought it was a measurement error until we realized that this is happening every 24 hours. We aligned it with weather data. that's independently measured to see that it was occurring around the arrival of photosynthetically active photons. And so then after discussing with some other biologists and soil scientists, we came to the conclusion that we're pretty confident it's a root exudate interaction. We've seen it across many different sites as well. It's stronger in healthier soils and weaker, so healthier organic or regenerative soils. It's weaker in conventionally farmed soils, or soils that are considered to have low biological activity that have been measured using other approaches like CO2 burst tests. And so we're confident this is what we're observing here. It will take more analysis and studies to prove that conclusively, but we found the best explanation. It even correlates highly with the degree of intensity of those of the photosynthetically active radiation. On a cloudy day, you'll get less or none. Sometimes if there's clouds moving through, we'll see a double spike occur. So there's a number of things that point to this. I'd be happy to share separately, since I don't have it ready here, what this looks like across a number of different sites. I'd be very happy to get to your input on this, Dr. Michael, and to learn more about what we're doing and see how we can improve this. But yeah, it is certainly highly unusual. The explanation we've come to, due to the periodic regularity of it, is that it's triggered by the arrival of not sunlight, but enough photosynthetically active radiation that's triggering a response from this. We've also seen this aligns with the daily minimum of VOCs in the air. So VOCs spike right as this happens. We believe that's also from the beginning of photosynthesis occurring in the plants as the stomata open up in the leaves and we see some gas exchange. So there's a number of different separate data sets that we've measured that all point to this. We are also open to additional explanations, and if you have anything else you'd love to add, I'd love to hear it.
Yeah, I was going to open up, Dr. Michael, if you're still here, would love to hear your thoughts on that.
Hang on. Yes. So, yeah, I was just reacting to that one. Spike patterns are just in biology not really very common, right? Outside.
Absolutely not.
Electrophysical. That said, we've seen, and I go back about 20, 30 years ago when we looked at high resolution measurements in bioreactors, which were at the Hertz level and so forth, or even below that. You were actually able to track and in bioreactors, you obviously have asynchronous cultures. And so if you have a synchronous culture, for instance, from yeast or E. coli, you can actually track on the respiration rate the meiosis and the cell division, et cetera, et cetera. So it is not unusual that these kind of things are happening at that very, very small and high resolution, but we literally haven't really seen that. And I would probably say from an eddy flux perspective that it would be important to probably pair that up, possibly with a separate measurement technology that is high resolution, such as eddy flux. And I actually do have a site in mind where we can do that, where we have a massive amount of photosynthetic activity. So it would be useful to do that, because if this is really something that is happening, I'm not sure that a flux tower would actually grab that, just because of the geospatial summation that is happening as a calculation of that. But on the other side, I would have to say, yeah, no, I haven't seen something like this happening yet before. It would be a very awesome validation of the technology, quite frankly. A very unique positioning of the technology also.
Yeah, I was sure when we first saw the data streaming in from the first devices we put in the field for analysis that we were seeing an artifact of some strike on the sensor or something like that. We figured it was temperature driven. But after looking at all the other different types of data we gather from these devices, none of that aligned and explained it. And so we started seeking other explanations. The thing that really blows our mind, though, is if you look at these spikes, you'll see the concentration rises slowly, but then it plummets. And I still can't wrap my head around that, because this is faster than these chambers. The arbiter chambers are vented chambers. It's faster than the CO2. It's much faster than the CO2 can vent from the chamber. It is actual reabsorption to the soil, and sometimes we end up below where we started. So again, this is a device that we didn't build, frankly, to do this kind of. We were not trying to build a scientific instrument here. We were trying to build something to give farmers' understanding of soil health and net ecosystem exchange. We started picking this up. It's super interesting. I'm excited to see it. We have seen when we do soil level, we call soil level CO2 measurements, so outside of its chamber. And you'll see over here, if we look at the bottom image, you'll see a black box on the chamber and then a black box next to the chamber. Yeah, perfect. Thank you, Charlie. We'll see a very small spike on that correlates completely with this, so that there's definitely CO2 coming out of the soil, but it's this unique signature we get from the chamber that's fascinating. And then, of course, the fact that we're seeing distinctly that it's correlated with healthier soils. So I'd love to work on understanding this. We're up on the Arkansas ARPA-E site now with a couple of these arbiter devices, Dr. Michael. They were offline because of a storm for a few days, as you're probably aware, but they're back on, and we're
They should
really
be back
excited.
on since yesterday, 10 o'clock.
Yeah, exactly. Yeah, we saw our stuff come in exactly around then. We're looking to deploy on more eddy towers because, of course, calibration and validation is important. One thing I will emphasize that's important to say here is that we gather raw data with these arbiter devices. We then process that data into soil flux, and eventually, as we refine the machine learning, next net ecosystem exchange. That doesn't change the validity of the raw data we've gathered. It just changes the processing we do. So as we deploy against more calibration and validation sites with eddy towers and other very, very high quality instrumentation, we can always go back and improve our measurements. And we look forward to continuously calibrating and validating and improving this product for our customers over time, just to get them the best measurements possible. And again, we really designed this to get accurate, but to also be durable and compatible with cultivation practices, and frankly, repairable and affordable enough that they can genuinely go out into a cultivated farm. And so a key thing with the Agrology's products is we expect active farming around them. We know that they will occasionally get damaged, and we'll be there to fix them. And the cost of repairs and maintenance is included in the service contract, because it's really important that if you want to measure this stuff on active farms, that it can be part of an active farm. And what we've learned over multiple years of operating devices around active farms is that things are going to happen to them. And so we've built them so that they can be repaired and affordable, and that we have a business model that can support measurement on active farms. So any other questions about that, or anything you'd like to expand on, Dr. Michael? But yeah, it honestly, when we saw that, I was like, oh, our stuff isn't working. These spikes are some crazy thing. There's no way this is real. And it took several conversations with soil biologists and soil scientists to convince us that there was a biological basis.
Well, you certainly have a paper at your hands, if not anything, right?
Yeah, just not the time to write it yet, but we would love to work with some collaborators on that. Absolutely.
Awesome. Are there any other questions? Any other curiosities or comments from anyone else? Going once, going twice. All right. Well, with that, I think we'll wrap it up. Once again, thanks everyone for coming. Really appreciate it. Here are the contacts for Adam and Miguel. I'm sure they would love to hear from any and all of you. Also, I will follow up with an email to you directly with the recording and the slides, and also keep an eye out for future webinars. So we're going to be doing 5 more of these at least this year, and really looking forward to seeing you all then. So reach out if you'd like to talk more about agrology or any of these things. We're here. And again, thanks so much for your presence.